All right, good afternoon, everybody, and thank you for joining us for a fresh new year of Webinar Wednesdays with PKD Foundation Chief Scientific Officer, Dr. David Barron. We're so excited to be back in 2016. As usual, we're live tweeting throughout the event. Please be sure to follow us at @PKD Foundation and tweet us questions and comments using the hashtag Webinar Wednesdays. We'll add your questions to the queue to be answered at the end. During the presentation, don't forget to type your comments and questions into the chat panel on the right side of your screen to let us know if you have any thoughts or anything you want us to share. We will have a Q&A at the end to answer as many questions as we can get to. Please note that the PKD Foundation does not offer medical advice, and the information shared this afternoon is not intended to be a substitute for consultations with your healthcare professional. Care and treatment decisions related to your health must be made in consultation with your health professional team. We are recording this webinar and will archive the recording on our website within the next few days. Today, David will be talking about a recently published article from the online journal PLOS ONE titled Improved Structure and Function in Autosomal Recessive Polycystic Rat Kidneys with Renal Tubular Cell Therapy. And with that, I'll hand it over to David.
Well, Happy New Year to everybody, and we begin our Webinar Wednesdays anew. I thought I'd go on a slightly different topic that isn't explicitly about a particular therapy or about how the kidney works, but rather what I do at my desk much of the time, and that is to read through the scientific literature. So I wanted to give you an example of such an article, and I should say from the outset that these articles are often difficult for me to read and understand because they are very specific. They involve sub-disciplines of biomedical research that I may or may not be familiar with, but I wanted to give you an idea of how one might look at these articles and get what you need from them. So, the scientific literature is where the data really reside, and the primary source, in my opinion, is always best, but at best it's very difficult for the non-scientist. One of the reasons is that these journals, and PLOS ONE is a very unique, well, not so unique anymore, exception, is that you will likely have to pay $31.50, which is the going rate, to get a copy of an article in the scientific literature. You can go to a library, but very few libraries contain all the various journals that are available. So your best bet is to be on the faculty at a university, be a student, or otherwise have access to those types of libraries. I want to say that the World Wide Web is chock full of information, but is it faithful to the original source material? Many times you can look up at anything you want on the internet and find an opinion that, if you want, will coincide with yours, or maybe you want to look at the arguments for and against, but you have to be sure that what they're talking about is really based in fact. So that's one of the issues about the World Wide Web. And sometimes there isn't even an original source, that there's a claim made for a food supplement or some sort of therapy or exercise, but it's really in the mind of the originator, but it hasn't really been out in the literature. And if there is an original source, how do you decide whether it's reliable or not? Even scientific journals vary in quality, and you want to make sure that what you're reading is an article that's been peer-reviewed. And I'll talk about what peer review is soon. And one should know that science is ever-changing. It is not static. Yes, the sun will rise every morning, but in the areas of biomedical research, new science supersedes old science. Some old science is classic and it's not superseded. But the point of science is that they are hypotheses to be tested, and sometimes those hypotheses fail and you move on to a new idea. The other thing I learned during my academic phase of my career is that science is 99% sweat, and tears. And what do I mean about that? You're working in the lab. I've slept over in the lab for experiments that lasted 24 to 36 hours. And maybe 1% is inspiration. And those are the aha moments that scientists live for. And the vast majority of us, let's face it, are not Newton, Einstein, or Watson, Crick, and Franklin, but we all play a pyramid represent the scientific article. They're all required for the structure, but each block in and of themselves may only have meaning to the investigator that did the study and to his colleagues, his or her colleagues that were doing related type of research. And this is an analogy that some scientists may not completely agree with, but in the case of the pyramid, the aha moment is that last block that you can barely see that caps off the pyramid. And so the foundation of that pyramid, which is the 99% blood, sweat, and tears that I've been talking about, are what make that final capstone and brilliant discovery possible. And that's, of course, what we hope for in finding a new treatment or treatments or even a cure for PKD.
David, can you pause for a second? They're having trouble hearing, and I think I'm going to move the microphone connected into mine because I don't think it's working through yours. So let's just switch this here. Sorry, everyone. Okay.
How is this? Do I need to start over?
I don't think you need to start over. There was just a little echo, I think. So go ahead and keep going, and everybody let me know if you have any more trouble.
Sorry so I was talking about the pyramids, and most of the blocks are the 99% of blood, sweat, and tears, and the last block is the aha moment. And when I was in graduate school and early in my academic career, I'm almost sad to say the way it used to be was that libraries were not electronic. They contained journals that were bound. Typically, all the journals for a year were put in a bound volume, and the latest unbound journals that were just sent to the library were available for perusal. When I was a graduate student, I had to learn German, not to speak it, but to read it, because English, although still the predominant language of science when I was in graduate school, nevertheless, there were very significant articles that were published in German, French, and other languages as well. And in those days, when you wanted a copy of an article, you could either go to the journal, which for many investigators was difficult because they may have worked at a university or college which didn't have the funds to buy all the journals. So, for example, when I published an article, I would get requests for reprints, and it was great for my stamp collection because most of the reprint requests were from outside the United States, where they didn't have access to the journal. But it's a slow and inefficient process. And when I was really interested in looking up a new topic, I used a card catalog. I did not put in keywords to a computer. And also, when I was a graduate student, there weren't nearly as many journals as there are now. So it was a little bit easier to keep abreast of the latest literature. So the way it is now, of course, is the internet and search engines, which really streamline the way I look at literature. I can just enter in a few key terms, and out comes all sorts of references that may or may not be what I was looking for, but more often than not, I find the lead I need, and it's very efficient and very quick. So libraries have essentially become electronic, but as I mentioned earlier at the beginning of the talk, access is often restricted. You won't see the types of journals that I need to look at very often in a public library. But there's been a bit of a disruption. I think that's a word that's used in the internet these days, and that is open access journals. And the article that we've offered a link to is in a journal that is open access. Not only is it open access in that you can read it, but I can actually give you the link without worrying about violating any copyright restrictions. If you read on the front page of the article, it really tells you that you can freely reproduce, distribute, transmit, modify, build upon, or otherwise do whatever you want, basically, with the article. And to give them my seal of approval, I've chosen one of their articles for exactly that reason. However, open access doesn't mean poor quality at all. It means very good quality, and these journals are peer-reviewed. And peer review really means that other scientists, peers of the scientists that are publishing the article, review it without having their names revealed, and they point out areas that might be rewritten, experiments that might need to be redone, and either tell you we can publish it if you make those changes, or it's really not publishable in this particular journal. At the same time, there are these open access journals. There's been a huge proliferation of journals and publications over the last 20 or 30 years. Part of this has been driven by the pace of scientific advancement. It's also driven by academic pressure. Even in my day, the phrase publish or perish was meaningful, and it's even more meaningful now. Getting a tenure track position at a major university is far more difficult than it once was. So the downsides of all this literature is that science depends on reproducibility, and in the old days, quote unquote, scientists did try to reproduce important studies. But now, who can really keep up with it all? You're doing your own research. You don't want to spend your time trying to reproduce someone else's research. So this proliferation of science has led to some of the issues around peer review. It's hard to live with it, but there's a huge risk living without it, because some of the peer reviewers are asked so many times to review articles that they begin to have conflicts in keeping their own time to themselves so they themselves can do research rather than reviewing it all the time. So this is, to me, a recent exciting article, and Alexis read you the title. What's in a title? Well, the words are actually fairly simple. Improve structure and function. So that's pretty self-explanatory. It means that the structure of the kidney and the function of the kidney in autosomal recessive polycystic kidney disease, ARPKD, in this case, however, not in humans, but in rats,
my mic suddenly muted.
It was fine, and then everyone's saying he's gone. I don't know what happened. I promise we didn't touch a thing.
So how many minutes ago was that?
It just happened.
Okay.
I was just getting these.
So I'm talking about the title.
Yeah, we're back.
And clearly, it's about how to improve kidney structure and function in rats that have the equivalent of ARPKD in humans. And the means by which these investigators do it is with renal tubular cell therapy, which, as I mentioned, I'll describe because that's the point of this article. The bullet below that shows the citation, and to be honest, I have difficulties with this citation. It used to be that you had the name of the journal with a standard abbreviation, you had the volume, which in this case I believe is 10, and then in parentheses the month that it was published, so 7 would be July. But now you have these electronic reference numbers, and this is very different than how I used to reference articles. And next in the cover page is the list of authors, and I didn't bother to list what the superscripts are, but if you noticed after K.J. Kelly's name, there's the number 1, and that refers to her institution where she did the research. And you'll see that most of the investigators, but not all, are from that institution. But what's very important is that there's also an asterisk in superscript, and that means she's the corresponding author. And typically the corresponding author is the senior author, and many senior authors put themselves at the end of the list of authors, so you'll often find the senior author at the end of this list. That's to allow a junior author or a postdoctoral fellow to be first author, which helps him on his career. And you will see other footnotes that describe various affiliations, and you'll also see a footnote that is somewhat sad for all of us. Not somewhat, it is sad for the foundation, for Vincent Gattone, who was actually funded as a core unit by the foundation, who has since passed away, but contributed to the research that made this article possible. So how is a research article organized? Virtually all articles will contain an abstract, and I think we all know what that means. It's really a paragraph that describes what the article's about, what the results are, and a sentence or two about what it all means. And then you get into the meat of the article, and it begins with an introduction, which typically is why they're doing the experiment, what they hope to get out of it. And I'll go into these various pieces of a typical scientific article in more detail. But the materials and methods for some who read articles is the most important piece. Is their rat model an appropriate model? Is the methodology they used legitimate in the eyes of the reader? This is what the peer reviewers are also looking for. And then, of course, the results section is critical as well. And you look at these results, are they plausible? Are the statistical analyses of the results plausible? And then the discussion, and some people think that this may be the least important part of an article because it's the author's speculation on what they think of their work. And often in the discussion, the best discussions are self-critical. They explain the shortcomings of the study, what needs to be done as a follow-up to the study. Others tend to actually say how great they think their results really were. Then there's supporting information. This wasn't common when I was in graduate school, but sometimes the very detailed information about the materials and methods may be included in supplementary information that you can't get in the printed journal, but that you can find on the internet under that journal's website. And of course, there are the acknowledgments, and these are very important. You want to acknowledge the support you have from other scientists that may not have merited being co-authors, but were nevertheless critical to being able to do the research. And then the references where you acknowledge all those blocks that build the pyramid that helped you design, interpret, and publish your particular contribution, which more typically is another block a little further up the pyramid, but not usually at the top. And just so you know, not all articles are organized in this way. Many electronic journals have played with this type of organization, but these are the elements that you should see in a typical research article. So how do you read them? I've alluded to this a bit. It's personal. The abstract, well, that's the first place to go to say, this really doesn't have anything that I'm really interested in. So the title looked interesting, but in reading the abstract a couple sentences in, I'll maybe put it on a reference list, but I'm really not going to go further. Then I might look at the introduction if I don't understand this area of research to get an idea of where they're coming from. But really, I try to look at the materials and methods, as I mentioned, how did they do the experiments, and can I count on the results as being plausible and reproducible? And then, of course, the results themselves. And the discussion, again, is sort of like the introduction. It's not critical, but I want to look at their impression of their data, where some of the issues may be still open to alternative explanations. Excuse me. And then when I was publishing actively, did they forget to cite your paper? And then you have sort of an insult to your ego, and you write them and say, why didn't you cite my paper in your reference list? But that's sort of neither here nor there. So the abstract. In this particular case, it was a very good abstract. The first sentence, why it's important to study ARPKD. The second sentence is really the statement of their first hypothesis that they want to test. And then the third and fourth sentences are the results. The fifth sentence is the statement of the second hypothesis, and I'll get to these hypotheses. Sixth sentence are the results based on that hypothesis. And finally, the seventh sentence are the conclusions and future speculation. So that's really what you want to see in an abstract. In the introduction, they describe what ARPKD is, because people may read this article that aren't in PKD research at all, but are interested in other aspects of this paper. For example, the concept of renal tubular cell therapy, but they may not be experts about ARPKD at all. And a certain amount of how the goals and experiments were achieved is included in the introduction. And there's also some description of why this work is unique and the inherent advantages of the approach they used and of the results they've taken. So, in materials and methods, the questions with what, namely rats, where from, and I'll get into that in just a bit, and how, what is the experimental design? And materials and methods are often very dense, and if you're not actively doing those kind of experiments yourself, they may sound rather foreign. But in this case, they use primary renal tubular cells. They describe how they got them. They use PCK and SD rats from companies called Charles River and Harlan. Now, PCK are polycystic kidney model rats that mimic ARPKD, and they're from Charles River. SD stands for Sprague-Dawley, and this is one of the most common white rats. When you think of a white rat in biologic research, it's more than likely a Sprague-Dawley rat. So for reasons which will become apparent, they only used female ARPKD rats, and they infused them with only male Sprague-Dawley rat renal tubular cells. And that was addressed to hypothesis number one. What would happen when you put into a PCK rat renal tubular cells from a normal rat? The second hypothesis is that these same female PCK rats were infused with something called an exosome. In my day, we called these multivesicular bodies, and we didn't really know what they were. But these multivesicular bodies open up at the surface of the cell and release these very small membrane-bound vesicles called exosomes. And these, as it turns out, contain very important materials. They may contain a gene, and meaning messenger RNA for a gene. They may contain a protein, such as polycystin 1, which is the mutated protein in autosomal dominant PKD. And they may contain other microRNAs. So the whole field of exosomes is something that is new in the past couple of years and really fascinating. So the second hypothesis is, okay, we're testing whole cells. Can we get the same results just using these little budded vesicles from these whole cells? And how does that work? And how do we know if our experiments are actually working? And then the materials and methods go a lot into how they measure the cyst size in both the control and treatment groups. In other words, some of the female PCK rats received sham infusions of cells. In other words, they didn't really get the cells, but the needle was still put into their tail vein, and some got the actual cells. So you want to know in experiments how the controls were designed and how the treatments were designed. And then you learn something about, the experimental design, but you don't actually see the hypotheses that are being tested laid out in the materials and methods. Those come often in the results. And this is a matter of writing style and how the journals are designed. So there are multiple experiments being done, and you have to look to the results for an experiment they did with ischemia. And ischemia means depriving the kidney, in this case, of oxygenated blood, which causes cell damage and causes cell proliferation to repair that damage. So that was a key part of their experiment. And I know I'm probably talking in riddles right about now, but I just want to give you an idea of how difficult it is even for other scientists to dissect some of the literature. And that's, of course, the bullet in my next slide, which is that it can be very difficult to understand. For example, I was wondering, what is SAA? Well, it turns out that SAA stands for serum amyloid A, and why is it used here? And this is where reading scientific articles become very difficult. If you want to know why SAA was used, well, go to reference 17 and learn why. And what does that mean? You've got to go back to the computer, you've got to go track down that particular journal, and you have to read that reference to find out why. It turns out that serum amyloid A causes cell proliferation. It's called an acute response protein, and they were trying to get kidneys in these female animals to be proliferating. They used plasmids. Plasmids are structures that contain a DNA sequence. For example, the sequence for serum amyloid A, in other words, the directions on how to make that protein. Well, how did they make the plasmids? You go to reference 17 again, and then you got to go to reference 20 in probably another journal. How was cystic change measured? Well, you've got to go back to references 23 and 24 and learn how. This is, of course, assuming that you're really critically reading the article and you want to make sure that everything they do is justified. And then they use FISH. You get stuck with a lot of acronyms in reading these articles, which stands for fluorescent in situ hybridization. And this is where it gets very clever as to why they used normal males and PKD females, because they were able to use FISH to identify the Y chromosome of the male cells from the normal animals that were infused into the female cells. You would not expect to see the Y chromosome in female rats, so that they knew after they injected the cells, if the cells survived, they could identify them by using this fluorescent in situ hybridization method, which means when you look under the microscope that you can see the cells with the Y chromosome light up. So again, very clever and described once again in reference 20. And then the conclusion, most articles are not standalone entities. So you will see lots of references that explain where they got many of these ideas and give credit to those prior researchers. So what are the results? This is what the reader's typically waiting for. Do the infused normal renal tubule cells improve the phenotype of PCK female rats? Is the experiment more effective with the cells being infused from these normal male rats also having the gene for serum amyloid A? these are all hypotheses. Do exosomes that I just described containing the message for PKHD1, which is actually the gene that is mutated in autosomal recessive polycystic kidney disease, get incorporated in and expressed by renal tubules in these ARPKD rats? So there are really 3 questions, maybe more, and these are all in the form of hypotheses. And so these 3 sub-bullets are the hypotheses. And I've already answered, I've given away the bonus question, but now you know why the donor cells are male and the PCK rats are female, because that's the way you can keep track of these male cells, which are essentially they're using cells as a drug or a therapy rather than a drug. And the only way to know if it's working is to be able to identify those cells, and the unique way that they're doing it is by being able to track those cells because they have the Y gene in them. So the discussion is, what do the authors think of their work? And to the point, their sentence in the discussion that I've quoted is that accordingly, we suggest that IRCT, another acronym, which stands for intravenous renal cell transplantation, can be applied to limit untreatable polycystic kidney disease. Now that to me is very exciting, and that's why I really tried to read this article very carefully, because what's also exciting to me is that these cells, which are foreign to the females, they're male cells, the immune system doesn't seem to track them down and kill them, so that these cells survive in the female bodies. The immune system doesn't go track them down like it would if there were a kidney transplant, for example. And that also brings me to the second point that I thought was very valuable in their discussion, and I'll quote, "Furthermore, multiple patients can potentially receive cells from a single donor." In other words, if you're only transfusing cells and not transfusing a whole kidney, one donated kidney or one kidney from a non-living donor can be used potentially to treat 10 or 20 or more people in need of a therapy for their polycystic kidney disease. So I think this work, although it may be many years until we see such types of studies in patients with PKD, really seems possible. And some of this work is being done with stem cells in other disease entities. So I think this is really exciting, and this is what I spend a lot of my time doing, because we at the foundation want to support the type of research that can really lead to potential therapies for this disease. So the discussion should typically end in a conclusion, and again I quote, "In conclusion, we suggest that in PKD, adult renal epithelial cells and exosomes offer a novel, physiological, and effective means to deliver normal genes. These potential therapies affect preservation of renal structure and function and limit cyst formation and expansion in experimental PKD." Well, I consider that quite an outcome, and I sincerely hope that through the NIH or perhaps the foundation or perhaps the persistence of these investigators and other investigators who think of their own experiments based on this significant block of research in that pyramid, really leads to an effective therapy for polycystic kidney disease. So I think this was an exciting article that's appeared just this past year, and I'm really going to keep track of these authors to see how they follow up, because it turns out that they've done previous experiments for diabetic renal disease in which this same sort of strategy has worked as well. So I think that this really has some real possibilities as we move from what we think of as therapies as being drugs to what we think of as being therapies that might be stem cells, or in this case, kidney cells from donors that could actually help forestall end-stage renal disease in patients with PKD. So in a postscript, I hope you can tell that I'm pretty excited about this research, but there are lots of questions and research that remains, and I think that's why some of us become scientists. For example, I mentioned, how do these infused cells escape the recipient's immune system? That's very exciting, but I don't know that the answer is there, but I have to do some more literature searching. The answer may be out there already, but it's not known to me. I could call or write to the corresponding author. That's another approach. And is the research translational? I think some of you have heard of the term translational medicine, and that's really what we want basic research to be in the biomedical area. Will it work in humans? I mean, we're not in the business of curing PKD rats. We're in the business of finding cures and therapies for humans. So it's important that we can translate this important type of research into the human arena. And is it practical? And will one infusion suffice in humans? Remember that we live many, many more years than the typical lifespan of the laboratory rat, which is only about 2 years. And that's with a lot of loving care. The rats of New York don't live nearly as long, although maybe a few do. So nevertheless, compared with the relatively recent past, the potential for an effective treatment for ARPKD and ADPKD has grown substantially. And it's partly by innovative research like this that needs next to be translated into potential human therapeutic approaches. And what's interesting about this model in the rat is, although it's the ARPKD gene, the phenotype, or what the kidneys look like, is the same as what they look like in ADPKD. So that I think that this is exciting work, not just for patients with the autosomal recessive form of the disease, but is also important for those with the autosomal dominant form of the disease. And the PKD Foundation, of course, is proud to provide grant support to innovators in PKD research, and that's what we have just recently done the beginning of this year and are planning to do year after year. So there are upcoming education events. My webinars, of course, will continue from month to month, and I want you all to know about the National Convention in Orlando, June 24th through the 26th, and there'll be special sessions for parents with children who have ARPKD and ADPKD. I think we missed a P. And there will be sessions and outings for teens and young adults with PKD. I think this is important. I'm a father. I have PKD. I don't know if my children have it, but children are notorious for not taking medication and not wanting to think that they think they're immortal, basically. So I think this is important. And of course, it's at Disney World, so what's there to lose? So please stay tuned to the org site for updates on the convention. And with that, I'd be happy to answer any questions you may have.
So go ahead, if you have questions, please submit them via that chat panel. You can tweet them at us. And I have a question while we're waiting for others to gather their thoughts. So what I want to know is what would the next steps be for these researchers? So they found this really intriguing conclusion. So what should they do next? What do we hope they do next?
So I think one of the next steps would be to see if exosomes alone can be as effective as whole cells, because exosomes are even less likely to be recognized by the immune system. I mentioned ischemia. I didn't give you the results there, but they were trying to push their model to be more proliferative, to be a greater challenge for the infused normal cells to actually have an effect. So they, if you read through the paper, they've probably suggested and are already working on successor experiments. But the real next step is to look at the plausibility of doing this in human beings, and that would ultimately involve the FDA and doing a pilot study, perhaps in people who are normal, receiving normal cells, kidney cells from a donor. Typically this would be a non-living donor, to see if those cells are destroyed by the immune system or they are under the radar like they appear to be in rats.
Okay. And again, if you have questions, let us know. And I will say, here we go. So we have someone, this is actually a really good question. They're wondering about the importance of replication of results. Why is that a thing?
So replication of results has a long, long history. And I'll start way back with Gregor Mendel, who was really the father of modern genetics. Even though he didn't know what a gene was, he didn't know what DNA was. He was a father. I think he was a friar, actually. And he was a gardener who realized that when he mated a plant that made yellow peas with a plant that made green peas, that all the daughter plants were green, for example. And then he looked at green wrinkled versus green smooth, yellow wrinkled versus yellow smooth. Some of those gene, or those traits as he would call them, were dominant, and some were recessive, which is exactly where autosomal dominant and autosomal recessive comes from. And if you do the experiments, a dominant gene should show 50-50 in the offspring. A recessive gene mating, where each parent has one normal gene and one recessive gene, should lead to 25% without the disease, 50% with the gene for the disease but no disease, and 25% that have both recessive genes and have the disease. So those two patterns are what happens in ARPKD and ADPKD. 25-50-25 for AR and 50-50 for AD. But when he counted his P's, sometimes it wasn't 25, it was 27.5 or 31, sometimes it was 22. And back in those days, he said, well, I'm going to clean this up and I'm going to make it 25-50-25, you know, because that's what he thought it should be, and that's what it is. But statistically, some families will have, with autosomal dominant PKD, will have 2 children with it. Some families will gratefully have 2 children who avoided the disease. So reproducing Mendel's work is technically impossible because he fudged the data. He got the conclusions right, and I'm not saying he was fraudulent in any way. That was just how an innovative botanist at the time made one of the bigger discoveries in biology. These days, reproducibility is an issue because you're too busy doing your own research, and often the work that is done is so specialized that to be able to reproduce, for example, the work that we discussed today would really require another laboratory to drop what they were doing and develop all these specialized techniques and plasmids and exosomal preparations to be able to reproduce the work. In other words, studies compared to 30, 40 years ago have become so sophisticated and complicated, it is very difficult for anyone else to do the research. The most you can hope for is that the authors, in this case, have done the same experiment multiple times and reproduce their results before they decide to publish. And that's my expectation when I read these articles. There are some cases, however, when that has not been done, and you find out, unfortunately, that exciting results can't be reproduced. And it's an image that the FDA and the NIH, it's an issue that the FDA and the NIH are dealing with as we speak.
So if I'm understanding correctly, what you're saying is it's on the researcher themselves to actually run through these experiments multiple times to ensure that the results are accurate and reproducible.
Absolutely. So just like I mentioned that the first study in humans is often done in just a handful of volunteers, I imagine that the studies that they did in this paper were initially just with a couple of rats to see if it was even feasible. And then they did another study with a larger number of rats to see if they basically reproduced what they did in the pilot study. And then they may have replicated the experiment several times so that they could do a statistical analysis and see if there truly was a difference between the treated groups and the untreated groups. So I count on that in the literature, but there's a lot of pressure on investigators. Money is tight. So you do, for important papers in particular, really have to comb through the fine details of how they did the work and if you think it's plausible.
All right. Well, there's no more questions coming through, and I think we've done a pretty good job of covering this one. So unless you have anything final to add, I think we'll wrap it up for the day.
So I guess I would add, I would like to think that the reason there are so few questions is that everything was perfectly understood, but I have the feeling that that's not the case. And I'm not expecting that you all go away with a perfect understanding of this article, but that you do go away with a better understanding of what's behind what you may read about the disease and the type of research that goes into what you read about the disease. When you go to your physician and that physician outlines a course of treatment for you, it's built upon the foundation of this type of research. And to people who might tell you, oh, they're playing with rats, this doesn't have any relevance, why are we wasting our tax dollars on this type of research? I hope that the message that I got through is that our advances in medicine depend on this type of research. It's not required that you understand it all. I can't understand it all, but it's required nonetheless, and it's really the foundation for the advances we've made in the last decades with PKD research.
And I will just add that a lot of the research that the PKD Foundation does support is basic research, not this research, but other similar typed research. And so that is just a reminder as to why it's so important to continue to support the foundation financially and otherwise, and to keep in mind that all of this, does have a long game result, and we are working in multiple pathways to try to get to that result as quickly as possible. But I think with that, thank you for your time. Thank you, David. And if you have questions or if you are interested in learning more about this, just reach out to us. You can check out our website, and you can always reach out to us at education@pkdcure.org and let us know what you think. Look to your email in the next few days, and we will send you a survey about this webinar, as well as a link to the recorded archived webinar on the website. And please do take note of that and fill out the survey so we know what we can do to improve. And otherwise, have a great day, and we look forward to seeing you next month when we discuss, our topic next month is drug therapies, and we'll have a couple of guest speakers joining David to talk about trials that are starting up or in progress now for new drug therapies for PKD. Thanks so much, and have a great day.